Wow, amazing guys. Thank you. So to wrap up the night, we have three amazing talks. They're going to be short, highly compressed, high density. I'd like to introduce Michael Torres from VITA RNA. And we don't leave yet. We have three more talks. And the last one from Benji about pump science. Maybe it's not financial advice, but stick around.
Excellent. Well, Hey, everybody. My name is Michael Torres. I'm the co-founder and CSO of R10 Bio, which is a project that is funded through VitaDAO. So really excited to see you guys virtually. I'm in Chicago for a cancer conference called AACR. But I will be in Dubai shortly. So really excited to talk to you about what we're doing at VITA RNA. Okay, cool. So yeah, just a little bit by way of background. Let me just go ahead and jump right in. So we're developing a gene therapy approach for going after aging. And so I look forward to sharing more about this. This is initially funded through VitaDAO. So we put the project through, raised about 91K. And then as we generated data, we now then moved forward into an IPT. So there is a token called VITA RNA. We raised about 300K, and we're developing, moving this through the drug development, pathway. And so just by way of background for myself, I am a scientist by training, but I also co-founded a company called Recode Therapeutics, which is a genetic medicines company that's raised over 400 million from folks like OrbiMed, MPM, some of the biggest life science investors in the world, including Pfizer, Sanofi, Amgen, Bayer. There's 2 assets in the clinic, and I help move those forward. So again, very much in the traditional finance biotech build space. I'm also the CEO of a company called Crossbridge Bio, which is my ADC company. So that's antibody drug conjugates. We're VC backed, including a fund based in the UAE, Crescent Enterprises. So really, again, just highlighting that, I'm bringing all of that knowledge and expertise, successfully fundraising from some of the biggest investors in the world, building data technologies that, ultimately can create best-in-class drugs. And now I'm excited to apply that to, the aging space through collaboration with, VitaDAO, Bio, Molecule, and, hopefully some of the rest of the stakeholders there. So again, just give you high-level view of our technology and the data set that we've generated today. So just to get you guys up to speed, for those who are who have spent time in aging, this is a well-known figure called the hallmarks of aging. But if you pay attention to what's here in blue, which are the primary causes of aging, a lot of them are focused around DNA damage, genomic instability, acquiring mutations over time. And these things can then reduce, protein function and then, be really correlated with aging. And so, if there was one therapy that could address, multiple aspects of aging, including the primary ones, this could be a really important step change in how we can actually achieve, the slowing down or reversing of age. And I know you're about to hear a talk later in this session that we'll talk about other opportunities to do so. So in the reversing state or stopping aging. So again, really exciting. So the biology that we're going after is the fact that, again, mutations occur over time. And in particular, folks may have, be well aware of methylation clocks and how that's correlated with aging. But if you drill down one level lower, what happens, and this is, a paper that was recently published, but I have a nice little summary here, graphical summary, that these methylations on the particular DNA nucleotide cytosine can then make it susceptible to a process called deamination. And then in the case of a particular codon called a CGA codon, which we'll talk about in the next couple of slides, that can change that C to a T. And a TGA is now called a stop codon. And so a stop codon tells the translation machinery to stop. And so you don't make proteins, and then, you can have diseases that are caused by these nonsense mutations. And so what people are finding and have appreciated that these somatic mutations, and these are ones that are just acquired over time versus germline mutations, which are ones that you're born with, that they track with age. So the older you get, the more these mutations occur. And then importantly, these mutations also affect like neighboring areas as well. So it's not just like a single, methylation that changes, that can then make a, stop codon and that's it. There's actually like very broad effects. And again, this tracks with age. And so what R10 is specifically focused on, and I think this is a unique aspect that, to our knowledge, no one else is trying to do, is, as I mentioned, there's a amino acid, and the amino acids are, what proteins are made out of, that has a codon called CGA. And as I said in the previous slide, this cytosine can be frequently mutated, be methylated. Again, it's susceptible to all kinds of, genetic perturbations, and then it can cause this stop codon. But if you look on this graph on the right, these are just a subset of proteins that actually have these CGA codons. So the CGA is not used everywhere, but there's certain proteins where it is used. And if anybody's in the cancer space or rare disease space or focus in DNA damage, these are all very important proteins. P53 is one of the most important tumor suppressors. RB, is a retinoblastoma protein, is involved in, it's another tumor suppressor, but in cancer. And then if you look down, a lot of these are DNA damage proteins. And again, arginine, because that CGA codon is so susceptible to mutations, arginine's actually the most common nonsense mutation that can occur over every other type of nonsense mutation with different codons. So again, highlighting that arginine CGA codons is very unique. And so if you put the pieces together, these mutations occur over time. And then arginine mutations in particular in very important proteins. So if we had a way to suppress or stop the consequence of those arginine mutations happening, then we potentially could have a very broad impact across multiple cellular processes that could, be that are associated with aging, whether it's cancer, Alzheimer's, Parkinson's, premature aging diseases, like in the case of ATM deficiency, I could go on and on. So that's what we're really trying to do with R10 and R10 102, which again is the project that was funded by VitaDAO and the VITA RNA token. So a simple schematic here, right? So these mutations are acquired over time and in very important proteins like P53 as an example. And then you have broad reduced protein function. So if we could deliver something that could rescue that, in our case, this is an RNA-based medicine to then put back an arginine instead of that stop codon, which is depicted here with using, in this case, a well-known gene therapy vector. Then we could restore translation, keep all of these proteins up for longer, and then hopefully significantly slow down aging, again, because we're hitting many cellular processes. So this is what we've been funded to do with VITA RNA and R10 Bio. So again, we designed some vectors that can put back this arginine in place of a nonsense mutation. And so this is called a Western blot. So you're looking at proteins from cells. In this case, this is a cell line called CALU6. It's a cancer cell line that has these arginine mutations in P53. So as you can see here, with no transfection, you don't see any protein because again, it has these mutations that make it non-functional. They don't make a protein. But with some of our vector designs, and you see two here, in this box, we are able to rescue P53 and presumably any other nonsense mutation that's present. But in this case, we use P53 as a reporter. So now we have vectors. We've screened several vectors, but these are two that I'm highlighting in this talk that again says that, hey, if you put this in cells, it can rescue these arginine mutations. And so We then next thought about, okay, what's a good delivery vehicle? And there can be many different choices here, but we wanted to go to start with AAV9. And this table shows you that this gene therapy vector, which is an approved, several approved drugs, actually hits many different tissues. And this would be important if you're wanting to, suppress as many arginine mutations that occur in all different tissue types. And that's presumably what happens, but we're looking to study, like, does it happen in, cells that are highly proliferating, like, stem cells and other tissues like that. But again, this is a gene therapy vector that's been validated. There's clear paths to the clinic, whether it's FDA, EMA, other regulatory agencies. Gene therapy is an ideal vector for that. So again, with the funding, with the token launch, we use that money to then work with contract research labs to then build these viruses, encapsulate our lead vectors into viruses, make sure that they can get into cells, do what we want it to do, and then from there we can start working on the traditional sort of scale up, which includes manufacturing, bioanalytics, et cetera, to support a trial. So we'll get to that in a second. But again, we then took those vectors and put that into cells. So this is some data showing that. And again, all done through VitaDAO funding, which I think is really exciting for us. We've also filed 2 patents. I skipped over that earlier, but again, we're putting the money to good use here. And so this again is those cell lines, and now this is just 2 doses of a virus that expresses a protein called GFP that you can look in cells. So you can see at this dose, MOI of 10 times 10 to the 6, we see good delivery. So then we put in our vector that would make the arginine rescue. And as you can see here, this is now the virus delivered into cells, and R10-102 gave us the best amount of rescue. So again, we're excited that we can continue to move forward with now this R10-102, which is our lead candidate that we're taking forward. And so this is just some data showing in cells the expression levels of R10-102 versus 101. So as you saw on the last slide, R10-101 didn't have much protein expression. And then you look for the amount of drug that we put into cells, you can clearly see that at the highest MOI, that 10 times 10, 1 times 10 to the 6, we have the highest expression of the drug, and we have lower expression levels with R10-101, which again, a dose response is something that you like to see in traditional drug development. And so again, we're really excited to take that forward. So then, as I mentioned, we're working with manufacturers to start scaling up. So we continue to move that forward because you need more and more virus, and it needs to be higher quality and safe for human use. And so along that path, you can start doing studies in mice to get your PK, your things that you need to support like trials. So we just completed a preliminary safety study, a PK and safety study in mice. It's very preliminary, but we took our virus, scaled it up, cleaned it up, that would be safe for mice, dosed it, single dose, to let those mice go for 2 weeks. We had no adverse effects, no change in body weight. We're doing more and more studies with the mice. We have the tissues. We're going to start looking at the R10-102 levels in the cells. So that's underway. So again, this will help us start planning out what eventually our dosing would be for a non-GLP tox study, as an example, and ultimately our GLP tox study as well that could support a first-in-human trial. So we're moving these things forward. And yeah, this is some more data, just graphs showing that at least in heme, looking at various blood cells, we didn't see any negative impacts from that. So again, very early, but very promising that a data set that we can continue to build off and get to the clinic as soon as possible. So this is what we're working on next and right now, and are looking for additional funding to continue to move this forward. So we're, again, want to get our manufacturing done. We need to do a little bit more dose finding in mice to understand what our GLP tox study might look like. And then we'd ultimately want to run that. You need several assays to support those studies, particular GLP tox. And then, we'd like to start, a clinical trial as soon as possible. And so again, we're going to be, talking with, UAE, which seems to be very forward thinking about longevity trials. And in our case, we can actually go after premature aging diseases that have nonsense mutations as a way to then, kill two birds with one stone. We get, our drug into an indication that we could potentially get approval for, but then that could really help drive the aging forward. So again, like the aging and longevity part forward. So this is what we're working on right now. And then, this is just a high level sort of budget and timeline of moving things forward. And again, we're really excited to leverage all of the work that everybody's doing to, get regulators open to these types of trials, and then, assets, longevity drugs, and then also, some of the preclinical work like Lev and what others are doing that could be really helpful to build the case that these newer modalities could be really important for aging. So again, this has all been enabled by, VitaDAO, Biomolecule, Paul, Benji, all the great folks there. And we're excited to continue to leverage the interest from the community to build what could be a really exciting, actual aging intervention. So with that, I think that's it. So thank you very much for giving me some time to share what we're doing here at VITA RNA and R10 Bio. Cool.